and I got to do this. All right, now we're back. So we'll go back to where we left off here. So much for that. I'll be putting up a lot of that at my age. A lot of people, famous people I know that I grew up with, are going to be dying, unfortunately. But that's not. Moving on. So we're going to spend a lot of time going forward with the impact of capsules, because when we get into respiratory diseases, capsules are a big player. I mentioned that as we were leaving last time. I just wanted to reiterate the point, because the way we deal with invaders is we swallow them. And so, in order to, we immobilize them, and we swallow them. And maybe you'll get a picture of that when you're doing the immune aspect in AP2, maybe not. But effectively, that's what we do. Okay. And so, looking at microbiological, you probably were staining this week. That's probably, you did staining this week. You didn't. You were doing environmental sampling this week. Love that. Then I go out and stain those samples. See, they do it a little. I do it more accelerated, but they're my colleagues. Everybody does their own way. So capsules, many pathogenic bacteria, because they risk phagocytosis. Effectively, this is what we would begin to look at, different factors that we call virulence factors. Even though it doesn't mean virus. Virus kind of literally means poison, but any disease entity has virulence factors, and the more virulent it is, the nastier it is for us. So whether it spreads fast, whether it replicates rapidly, whether it causes a lot of damage in a short period of time, it's something called infectious dose. You only need a little exposure to get sick. Those are all virulence factors. We'll go over all those down the road. Again, phagocytosis. The other thing is biofilms, and the most famous one, of course, is dental plaque. Okay, so the bacteria basically stay in place. This is interesting, and this is impactful to nursing. The, the biggest single problem we had with what are called nosocomial infections, such an important word, I got to write it down. If you haven't heard it 63 times now. Nosocomial means hospital induced. Literally, it refers to an infection that you get, someone who did not have an infection in that system and gets an infection. That's a big problem. Okay, because insurance companies don't like to pay for it. We'll get to the business aspect of it. But the reality is that's a worry. And the number one area forever, and still probably number one, UTIs, and most of they come from catheters. Okay, because indwelling catheters, they tend to use more straight catheters now, and you'll, for all of you in the nursing sphere, are going to get a tremendous introduction to catheterization. Okay, but they colonize on, and that's a biofilm link. I mean, so it's infection looking to happen. That's one of the real problem with indwelling Foley catheters, which was the standard for a long time. Intrauterine devices, which I have no idea if they're popular or not as modes of contraception. At this point, pacemakers. My experience is with these other implanted medical devices, and those would be orthopedic implant devices, screws, artificial joints, metallic hardware, etc., etc. That kind of stuff. And it's a big problem because certain bacteria with that glycocalyx form a biofilm that's very difficult to penetrate. And part of your choice for antibiotics has to do with looking at those and finding the antibiotics that can find it from a physician's standpoint. But I mean, there's so many nuances, so many levels to that. That's where the micro aspect is interesting. It's not that necessarily you're going to have to deal with it, but to understand. Because for many of you people who say, why is this happening to me? At least you have an idea. You don't have to necessarily give a definitive answer, but you have an idea this may be it, or, you know, some form of interaction and communication would be useful. This is just showing you, and there's a zillion of these, how they colonize, they divide, they stick together, additional microbes kind of join the party. And you can see here's a catheter surface. Actual, they're doing it. This is not a representation. This is just an enhanced electron micrograph of all the different kinds of microorganisms, little flagella and things like that. It's pain. So there's a lot going on with that, and that's really what I think. To me, that's the single most important thing about what you're doing in microlab, because it's unlikely you're ever going to have to use the microscope again. I'm just saying. depending on where you work. Sometimes you may, depending on what your field is, you may do slides like that or help to prepare slides or take a quick look at them. But the reality is learning the technique, is learning aseptic technique, how to handle things to not contaminate them. That to me that's what that's all about. Getting used to sterile transfers or aseptic transfers, whatever you want to call them. Okay, it's not used for attachment. I don't care. I'm sorry, the flagellum is probably not an attachment. Two main types of bacteria. We're getting there. Gram-positive and Gram-mal. We're going to get there. A substance in the envelope, a structure of some bacteria can cause severe symptoms in humans, and that's where you get to the lipid A. And this is the big stuff coming here. So I love it when they refer to the cell envelope, as I said before. To me, an envelope means that it's a structure that surrounds it, that it's a unit function. So it's composed of all the structures outside of the cytoplasm, two or three basic layers, the cell wall, and depending on the complexity, the membrane itself, and then in the gram-negative bacteria, the outer membrane. So even though we have limited membership here today, this is a really important stuff. I'm so glad you're here. Oh, we have people coming. I'll wait till they get here. We waited. This is just me. Look at that. That is, and this is the, I'll show you another set of illustrations. These are the two types of cell walls that bacteria have, with, again, limited exceptions. Gram-positive, a very, very thick outer layer of this magical substances that you're going to see is called peptidoglycan. And the gram-negative, which has a comparatively thin amount of peptidoglycan, but has a membrane on the inside adjacent to really the cell's membrane, and this very interesting membrane on the outside, particularly the outermost layer. And that you'll see is a very, very famous lipopolysaccharide layer. And that's something, so there's two unique things here. Peptidoglycan, which we only find in bacteria, and the lipopolysaccharide, this pink depicted here outer layer, that is only found in gram-negative bacteria, another level of identification that's there, another level of uniqueness, if you will. So everything, so again, the single most important thing that you end up doing in lab, I think, other than the techniques that you're developing, is to understand the distinction between gram-negative and gram-positive. That's the first thing we do when we have an infection. We do a thing called a gram stain. It's a proper name, capital Gram. It's named for a guy who was a Danish scientist, microbiologist, who worked in the lab and felt that there were two different kinds of bacteria and had to put together. Had difficulty. He would stain, one would stain or the other would stain, but never both. And he had to figure out a technique to make both of them stain. And long story short, you'll get this in lab. The magic ingredient was an iodine compound. And what the iodine did was it took the first stain, which you'll learn about, called crystal violet, sometimes called gentian violet, and it allowed that stain to embed in the peptidoglycan in the gram-positive outer wall. Then he would introduce what's called a counter stain, first using an agent to wash away the existing stain. So in the gram-negative, it wouldn't embed because of the outer membrane, and then it would pick up the second stain. And so effectively pink and red became gram-negatives and blue or purple became gram-positives. And it's called a differential stain because you're using two contrasting stains. You'll do this a lot. The endospore stain is green and red, okay? The acid-fast stain is reddish and blue. So you use contrasting color. You wouldn't use orange and red because they're too close to one another. And there's a lot of staining differences between them. So that's the background information. So the first thing we do, first test that you do in the lab, when you get a sample from someone takes a swab at bedside, we call culture and sensitivity. So what you want to do, there's drainage coming from the wound or there's a wound itself. You take a sterile swab, it's got some stuff on it, put it in there. Maybe it goes into a test tube. Maybe it goes directly into we take it directly from the blood. It's called a blood culture. All of those things, very straightforward. It's just a technique that you learn how to do. And basically you take them to the lab and they put them on a plate and start to grow them with a certain technique that you also learn in lab called plating it out. And then you'll take some of that specimen and you'll gram stain it. And that's really what that does from a physician standpoint. When we would do that in the office or we'd send it to the hospital, which was a very short distance when I was in the office, we get a stat or an immediate gram stain that was done. And they would basically tell us, oh, you have gram positive clusters along with white blood cells. You got a staph infection. So you can treat it accordingly. You make a good guess as the antibiotic, and then you go from there. You have gram-negative rods and white blood cells. Okay, I got a gram-negative infection. You're going to be choosing a different antibiotic for the most part. So it gives you some information. You make what's called an empiric choice. So empiric means you have a certain amount of evidence based on the location. I mean, if that's drainage coming out of a catheter, or it's on a catheter, it's a urinary tract infection, you have a pretty good idea what antibiotics you're going to be choosing based on that. So you're not prescribing something in a vacuum, which is fraught with danger. Love using the word fraught. That's okay. By now you should know my fascination with this type of stuff. Little background information. Peptidoglycan, it looks like a giant block there. It's not solid, it's porous. Okay. So it's all about moving things in and out of cells is how they survive. You don't need passageways. Membranes are made out of fat. Water soluble things can't get through them. They need passageways. So you have porin proteins, read that as pores, here, and you have different types of protein passageways here. So you have pores, either they're aquaporins for water or materials can get through, and whatever peptidoglycan is limited, but it's still effectively porous. So these cells do not require having these additional porin proteins on or pores on the exterior because it can get through it. Gram positives have this chemical called teichoic acid that helps to secure it to the membrane. Here, the peptidoglycan is sandwiched between the inner membrane, which is a regular phospholipid bilayer, good to its mother, sends her a Christmas card every year, calls occasionally on her birthday. That's what it does. It's your standard, just like over here, embedded proteins, the usual stuff. That's there. Versus this bad boy. This has an inner leaflet that's phospholipid. You can see the little balls like you saw before in AP1. But on top, that different color tells you it's a different chemical. I'll show you a couple of illustrations that might help with regard to it. So, yes, they play a pivotal role in determining shape. Strong structural support. So, particularly, it is very resistant to osmotic pressure. That means excessive fluid. Again, with bacteria, you will see salting or dehydrating, sucking fluid out, distorts the relationship between the membrane and the wall, and the membrane ruptures, dies. But because of the cell wall a la a plant, excessive amounts of fluid doesn't bother bacteria. Go ahead, water me, see if I care. They live in water a lot of times. They're happy. Water a plant. I'm having a good day. I got one. It's like my dog when I take him to the camp bow wow. Thank you. I didn't get you anything. I'm sorry. I make the turn. He sees it. He goes, in the car. I'm having a party. That and when he can get into the cat litter. Snacker. You're snacking. So certain drugs target the cell wall, disrupting its integrity, causing it to rupture. And we're going to talk endlessly about cell wall antibiotics and its relative rigidity from peptidoglycan. They show you this, and this is a little bit complex about this. And no, you don't have to extensively know that it's L-alanine and D-glutamate and L-lysines and D-alanine in these what are called tetrapeptide chains. Don't have to. Here's the skinny on peptidoglycan. Peptidoglycan, as its name suggests, peptido means it has peptide bonds that are part of it. Glycan, that end, glyc is sugar. When you put an N at the end of it, glycans means they have nitrogen. Nitrogenated sugars are very good structural materials. We use them. Go back to early AP when you did the unit on tissues. and you'll see that they had glycosaminoglycans are in there that were the framework of a lot of that ground substance in connective tissues. Okay, nitrogenated carbohydrates. Insect shells are made out of one of these. Okay, they have nitrogenated carbohydrates. The tough part of a mushroom, if you like shiitakes, which I do, okay, you can't eat the stem. They're good for stock, okay, because they're too fibrous. It's because they have that material in them. So they're very, so it depends on its name is on the tip of my tongue, of course, and I'll remember it in a second. It'll come to me in the middle of the night. So what this has is alternating nitrogenated carbohydrates. They're called NAMs and NAGs. Why do we call them that? Because I don't want you to have to remember N-acetyl-uramic acid or N-acetyl-glucosamine. Glucosamine is a famous name in our bodies. Okay, you go to Costco, you go to Giant Eagle, you go anywhere, and you're going to see it where they have all the pharmacy stuff, glucosamine with MSM. Okay, you're going to see all of those kinds of things. Those are agents that people take as supplements for arthritis because they got to rebuild carbon. So you see all those things. So N-acetylglucosamine, we call it NAG. N-acetyluramic acid, we call it NAMs, and they alternate. That's what polymers do. So a polymer doesn't have to be like cellulose can be all glucose chained together. It can be more than one item. So it's repeating is NAM, NAG, NAM, NAG. Layers of it. They're like bricks. They're the structural stuff, but they're porous. They're all chained together in multiple layers. Now, if you are making a brick wall in your backyard and you just put brick, brick, brick like that, one on top of the other, it's strong, but it's not secure. I mean, if you push on it hard enough, it would fall down. There's nothing holding it together because it doesn't have, and so here's another layer and another layer, another layer. It doesn't have mortar. Peptides are the mortar. So they're like this. They look kind of like that. They're called tetrapeptide chains and peptide interbridges. And that's what you're seeing to some extent depicted here. NAMs and NAGs, okay? And these are, the interbridges are between them. The tetrapeptide just means there's 4 amino acids chained together. Like dipeptides, 2 amino acids, tripeptides, 3. It's not based on the number of bonds, it's based on the number of acids. And here you have the NAMs and the NAGs, and they're interconnected. So that's what gives it its structural rigidity. It's the combination of both. And when we target it with antibiotics, they either target the assembly of this or the attachment of these, whatever, it doesn't matter. Okay. If they target it and it doesn't let it be assembled, then the wall isn't very stable and it can go. Strong but flexible. Glycan sugar chains cross-linked by short peptide protein fragments. 20 to 80 nanometers, roughly 30 to 40 layers of peptidoglycan thick. They contain, and this is a marker, and it does help to attach things to it, teichoic and lipoteichoic acids. Maintenance and enlargement, acidic charge. When you get into staining, when I told you about the stains that we use for that Graham stain, one you're going to use called, you may have used it already in high school, methylene blue, very, very commonly used. Crystal violet, safranin, carbofuchsin. All of those stains that we commonly use in the laboratory are what are called basic stains. Basic does not mean elementary. Okay, basic means basic chemical charge. So they're basic in their chemistry. They happen to be positively charged. Acidic is the cell wall has acidic elements like those teichoic acids, and it's negatively charged. Positive, negatives attract. That's how stains work. Okay, I'm doing your lab for next week already. Tell Dr. Dress I got it handled. Tell Dr. Hilwick I got it handled. They're my friends. Don't. I'm not trying to usurp their authority. He also used usurp. I'm going to start writing down the words. You need to expand your vocabularies. My wife always surprised me because we're both sitting there looking for the best word. And every time I use grammar inappropriately, it's terrible. I had that at house too. My sister was a high school English teacher before she went into the business world. Thinness gives gram-negative cells more susceptibility to lysis. Probably true, but that outer membrane insulates them because not a lot of antibiotics can penetrate that outer membrane. That's the real problem. It has, depending on the author, it probably has one, maybe two sheets. It's a thin layer of peptidoglycan. And it's in the middle of these two membranes. It's a membrane sandwich with peptidoglycan and a liquid gel-like substance called periplasm in the middle, and a lot of action in the periplasm. Periplasm is where bacteria have the means when an antibiotic comes in, they can pump it back out. They can bring stuff in and process it within the periplasm is a busy area that's here. So, and that's an area that's understudied for a lot of things that are going on. So you'll see it here, and in there they're showing you the gram stain a little bit. So they give it kind of short shift, and so this PowerPoint from the Nestor textbook, okay, that I have for you, and you'll see it obviously. And I'll just scroll down until we get to that area because I think the illustrations are better. They're similar because they're both from McGraw-Hill. And so if you have one, they use the same kind of, and I see no reason not for you folks not to, take a good look at them. And this is just movement across membranes. Where my this is, and so there's their description, which doesn't look quite as good in this part. Gram stain, purples and pinks that you see over there. So I'll expand it. Now, peptidoglycan, NAMs and NAGs, peptide interbridges, here, tetrapeptide chains here. This is kind of where I got that little drawing. These are their actual names, etc. This is the chemistry. Certainly don't have to know it. Relatively thick, depends on who you read. Teichoic acids extend. Gel-like material below. And again, things can pass through just like in cytoplasm. This is a better illustration, I think, of the outer membrane, inner membrane peptidoglycan complex of the gram-negative cell. It's such an interesting cell, and they really give you a good look-see at this stuff. So again, normal plasma membrane, peptidoglycan, bridged together a little bit, pores and channels that allow fluids and small polar molecules to pass through. So porin proteins and aquaporins, whatever they call them. Now we get to this other layer that's there. We have this the external leaflet of the lipopolysaccharide layer. Okay, of the external layer of the outer membrane is the key. It has this. It's called lipid A. The lipid component, the internal component that bonds to the fatty portion of the internal leaflet of that outer membrane, is known as lipid A. Lipid A, by definition, is unique. That's the unique area of it. The lipid A is what is referred to as an endotoxin. Endo means it's inherently part of the structure. The way bacteria hurt you, whether it's tetanus, whether it's botulism, whether it's Staph aureus, which is MRSA, effectively, okay, they generate what are called exotoxins. Exotoxins are secreted. They're spit out of the bacteria. They're a waste product, if you want to think of it easily. All bacteria generate, for all intents and purposes, exotoxins. Endotoxins, if you see the word endotoxin, the answer has to be a gram-negative bacteria. Whether you see it on a test of mine or you see it on an NCLEX exam or a licensure exam, anywhere. It is an endotoxin. Have a gram-negative organism, probably use E. coli because it's sort of the one that everybody knows, is the answer. Because you only find it in gram-negatives. When a gram-negative organism, because of this, gets someplace where it shouldn't be, most of your internal compartments are sterile. They're not designed to have bacteria. So if you have a burst ulcer, if you have a tumor in an ovary and it bursts, If you have an ectopic pregnancy and it bursts, if you have a bowel cancer and it bursts, what's going to spit in there? A whole bunch of garbage that's filled with gram negatives, because all those tubes linings are generally covered in gram negative bacteria, particularly gastrointestinal and genitourinary. And what that means, that generates something called gram negative sepsis. And your body responds to that dramatically different than other things. It responds to this invader with almost like an allergic reaction, a la anaphylactic shock. We call it septic shock. Your immune system goes crazy, releases a ton of chemicals that open up blood vessels to allow white blood cells out. Your blood pressure drops, your heart rate elevates, your blood flow to vital structures, kidneys and things like that stops. You're what we say in medicine, we use a scientific term in medicine. You're in big ass trouble. It's very scientific. You're up the proverbial creek. You know what I'm talking about. Another great TV show, Schitt's Creek. If you didn't get a chance to go watch that, it's hysterical. Nevertheless, that's the idea. Famous people die of that all the time because something ruptures in that. That's a very, very common problem when people have, again, genitourinary cancers. That's one of the things that sort of goes behind it. That was me. Is that you or me? Yeah. That's a term I think. And all of that comes from that. So understand you can have a bacteria like E. coli, one of those killer strains of E. coli, which you'll learn about, called O157H7. That is produces a very deadly exotoxin, but it's also an endotoxin. So it's not limited. And for whatever reason, students get confused about that. So all bacteria generally, certainly ones that hurt us, produce toxins, mostly exotoxins. Gram negatives, by definition, always have an endotoxin. If it gets somewhere where it shouldn't in sufficient number, it can cause damage. That's the lipid A. Protruding, it has a series of chains of sugars. They are identification sugars, just like sugars that we find, say, in the ABO blood group. They're markers that protrude from cells. We use those to type. So they're called the O antigen. So that E. coli, that's O157, has a specific chain that can be measured with molecular biology identification techniques. And that's how so whenever you see E. coli with an O prefix and a number, that's how they identify it. It doesn't speak to it unless the number corresponds to a strain that has particular properties. It could be 06, 023, whatever it is. So that O157 is a specific identification marker. The H7 has to do with a flagellar antigen that's also used to identify it. So that's where they come from. We use these all the time. When we get to viruses, you're going to hear about H and N groups. So when you hear about flu epidemic, you hear about H1N1 and things like that. And if you didn't, you will. And they're also markers. They stand for two other chemicals called hemagglutinin and neuraminidase. Such fun. You thought this was going to be just a quiet Friday at the ranch. Okay. Outside layer, the LPS layer, signals the immune system to invasion by gram negative. Small levels elicit response to eliminate. So if it's a tiny amount, and we get this all the time, our bodies are constantly alerted to things that are coming in. Most of the time you don't know about them. How many times have you said to yourself, or someone has said to you, I feel lousy today, and the next day, how you doing? I heard you weren't feeling so good. I feel great. That's your immune system just helped you. It dealt with a problem relatively expeditiously today. Relatively expeditiously, and you feel better. So you didn't manifest I don't know, I'm in one of those crazy moods. You're always in that. So the LPS is called an endotoxin, large amounts. Bloodstream can be deadly. Lipid A is the trigger. The O is identification. Outer membrane blocks passage of many molecules, particularly antimicrobials. Unfortunately, again, the player that doesn't work very well with that are penicillins. And you'll find out down the road that penicillins and its relatives are our most reliable antibiotics other than their allergy profile. The periplasm is that space in between things can get in, things can get shipped out. There's a variety of transport mechanisms that are part of it. So here's the penicillin nugget. Peptidoglycan synthesis doesn't allow those chains to cross-link. More effective gram-positive and gram-negative. The outer membrane of the gram-negative blocks it. That's when we get into that. I'm just sort of preparing the way for understanding what antibiotics you're going to see, what are called first, second, and third generation, or what are called semi-synthetic antibiotics. Antibiotics are naturally occurring, and we find them in nature. Take them to the laboratory, we tweak them chemically. And then they have a new name. They're called semi-synthetics because they're partially made or modified in the library. And this is terminology that you're going to become familiar with. Many of you in the nursing sphere, when you get that, you're going to hear those names. And that's why I'm trying to give them to you. Lysozyme is something we see inside lysosomes, but it's in a lot of our secretions, tears, saliva, other body fluids. It specifically targets peptidoglycans, part of our inherent defenses. We talk more about that, or you shall, when you do the immune chapter or chapters in AP2. So let's see. And that's now we're going back. Let me go back to the other PowerPoint and see where we're going. So I kind of go between them. It's fun. So, interesting. Acid-fast bacteria. This is a stain you probably try to do in lab. There are some interesting techniques. We actually have one that works. I'll have to make a mental note and tell Doctors Hillwig and Dress about this if they do that stain. Remember, I wrote it before that in the last class, that mycobacteria. Another one called nocardia, which we don't see a lot of. They contain peptidoglycan. By definition, they're gram-positives, but the fats in the cell wall are so dense, they actually become waxy. Very long chains. By definition, acid-fast means the stain fastens itself. Remember we talked about carbon fixation and nitrogen fixation, okay? Meaning that doesn't mean to fix in the sense of repair. It means to affix, to attach. So acid-fast means you fasten to that acidic moiety, which is called mycolic acid, and is a big player because very little in the way of only antibiotics that tend to get through that tend to be nasty. And of course, they're used because there isn't a whole lot of leprosy going around, mostly for tuberculosis. Tuberculosis, unfortunately, a lot of the antibiosis, the approach to treating it, can be as harmful as the disease itself. Most people have to be on multiple antibiotics because it tends to mutate relatively rapidly as it's growing because they're genetically unstable. So most people, if you know somebody, it's rare that they're on one antibiotic, and they do cause a lot of side effects. I had a student 16 years ago I was teaching at Chatham, and that was UPMC Shadyside School of Nursing students, which I did almost all the micro for. And she would alternate either getting an A or not doing particularly well on a test. And one day I said, "What's going on? Do you only study every other test?" And she goes, "No, I take them." Took care of my aunt for a long time. She had tuberculosis. I was exposed. They put me on an anti-tuberculosis drug, one called isoniazid. And some days I just can't think. When you're not feeling like that, was you ever think of telling me? And maybe you could take it another time. Oh, can I do that? Yeah. I give up. Okay. Non-typical cell walls. This is the archaea. Very distinctive. I really do not have the ability or the knowledge to comment on them. We just don't see them. So they're there. To me, the only question is, where do they exist in extreme environments? And they don't contain peptidoglycan. Those are the two things for time being. Mycoplasma. I talked about this a little bit. They have elements of peptidoglycan. They don't have a cell wall. They utilize sterols, chemically similar to the cholesterol in our membranes, to stabilize. But the big player in this, and we'll explore this in Pneumonias is mycoplasma pneumonia. They use the E at the end of it. Walking pneumonia is the classic definition. Atypical pneumonia, because it is not as severe. Most people with pneumonia are very, very sick. You've got a fever, you can't get out of bed, you can hardly breathe. Here you have mild fever, you're able to function, cough that lingers, so it takes longer to go away, but you're much more functional. Okay, and there are some oddball things. We see a lot of persistent infections, and even for things that sometimes things we're immunized for. We'll talk a little bit about whooping cough, which is called pertussis. A lot of people get that long term. So there are a lot of things that resist antibiotics and are very persistent, not necessarily life threatening, but very annoying. And potential, and a lot of, and you'll see a lot of this course, we emphasize something that was emphasized endlessly during COVID: comorbidities. And that's when people are sick, they're much more susceptible to problems. If they have underlying diseases that compromise their immune system for whatever reason. That's why I treated diabetics. I don't know, 65 to 75% of my population were older diabetic patients. They get sick easy. Take a long time to get better. Okay, gram-negative outer membrane, specialized. There's the lipopolysaccharide signaling molecules and receptors. It's an endotoxin. Special membrane channels that allow things to get through it, because remember, it's not inherently porous. And so within it, the lipid bilayer with proteins embedded, energy reactions, nutrient processing, synthesis, transportation, it's selectively permeable. So effectively, it does all the things of a membrane. It's just structurally a bit different. The outer membrane of the gram-negative contributes an extra barrier. And again, to underscore it, resistant to certain antimicrobials. I hate painting with broad brush. They've modified this PowerPoint. And again, I emphasize PowerPoints are made by people who are not scientists. They're not the author. They're made by graphic designers that are hired by whoever the publisher is, in this case, McGraw-Hill. They're not always, sometimes they're humorously inaccurate. Okay? So, I found some very unusual things in them. I endeavor to point them out. Don't go to an instructor and say, it was on the PowerPoint. Because they're going to say, well, look in the book. Now, if it's in the book and it's a misprint, it's a different story. PowerPoint alone is not it. Okay. I don't know that they're more difficult to inhibit or kill than gram-positive bacteria. I just think there's a little less tools. Are there less antibiotic choices? Maybe. I don't know that that's necessarily true. Alcohol compounds dissolve lipids in the outer membrane and therefore damage the cell. Okay. I'll buy that in a way that alcohol disrupts membranes. That's one of the way alcohols work. Alcohol is, you're going to be taught to give injections, wiping it down with alcohol. If you're taking a multi-injectable vial, which they don't use much, they used to, and you wipe the top of the vial, put the syringe in, blow in an equal amount of air. I've given a few thousand injections in my day. Okay, pop that in. Okay, after you cleanse it off, that alcohol is getting rid of the surface dirt. This is not sterilized. Soap and water would work. Plain water would work. Alcohol evaporates in 15 seconds. There's plenty of bacteria on you. Again, you're instructed to do this in clinicals. Do not say, Dr. Stein said this really doesn't do shit. Just look at it. Yes. Okay, I'll do that. Thank you for pointing that out. Appreciate it. Prompt, perky, positive, prepared, all those P's, patient. So alcohol, okay. The ones on the surface are normally on the surface. We get rid of them. So extra barrier, 7 structures in the cytoplasm. Okay. And then we'll get to endospores. A point of information. That whole gram-positive, gram-negative thing, there's going to be a ton of questions on the first exam. That's a major point. Effectively, we dedicate almost a whole lecture to that. So, Be advised, be warned. Harbinger. I want to reiterate that despite my extensive vocabulary, that in my house I was third in Scrabble. My sister, the English teacher, second. My mom never went to college first. But a very bright graduate of Weequahic High School, Newark, New Jersey. 70 to 80% water is the cytoplasm, just like regular cytoplasm. All the stuff's in there, just like our cytoplasm. The big deal is here. Bacterial chromosomes. I will show you something. It's a good way to, and I should have it. This is the chromosome number. Let's see if I have it here. That's the Google Meet thing. I wonder if I got, it's one of these animations. Okay. So I cooked it up, chromosomal compaction. Don't you know? These animations are generally very good. Okay. And what's interesting about that, without getting into the loop domains and anything else, he uses the classic analogy, which is what they would call a gum band around here. If you're in touch with your inner yinzer, I married two girls from Pittsburgh, not at the same time. Did not. And you just get, it's like you have one of those planes with the, remember? Rubber band plane. Am I too old? Are you all too old? Remember that? And the thing would kink up. That's politically correct term today. I don't know. Nevertheless, that's effectively what happens. So as we get back and look at the PowerPoint part of it, you have a tremendous amount of DNA. Again, disproportionately a large amount for a very small area. So it undergoes this supercoiling. So it's very interesting in that regard. So there's a lot of DNA. And what's interesting down the road is when a bacteria dies, it explodes out. And you'll see a number of illustrations. And when it explodes out, it fragments. It begins to deteriorate. Those fragments can be absorbed by other bacteria or carried to other bacteria. And that's one of the ways bacteria mutate. So it's another interesting, and they're facilitated by these tiny circles of DNA that are useful but non-essential, typically about 20 to 50 gene units around, and they're circular for the most part. They're called plasmids. And most bacteria have them, and they can come and go. And under undesirable conditions, they'll actually form additional plasmids. When the conditions improve, they'll actually go away that are there. And plasmids can move by that, when we'll get to this when we do the genetics, that F or fertility or sex pilus from one bacteria to another. That's another way that we see genetic mutation. That's probably the major way for MRSA development. So there's a lot there. So when we say useful but non-essential, again, the word essential means has to have it to survive. And that is not the case with plasmid. Confer protective traits such as drug resistance, which is very often where the MRSA comes from, various things. And we'll see some examples of toxin and enzyme productions that go along. Because to some extent, that mutation is what changes a bacteria from being benign to being deadly. So the hereditary material of the most bacteria exists in the typical bacterial chromosome. Plasmids not essential. Plasmids can be attached to it, but less frequently than they are separate. They call it the nucleoid this because there's no membrane. So it looks like a nucleus, but the reality is it's not sequestered inside of a membraned area that are there. So that's all part of it. We see some other things. I'll show you. We won't have the time today to show you those other illustrations. And then lastly, ribosomes. Ribosomes are protein synthesis. The difference, and they're mostly ribosomal RNA and protein. In bacteria, and we're going to get into the distinction between eukaryotic cells and bacterial cells, it has to do, and I commented on before, the 70S ribosome. We see it in bacteria and archaea. You see and it's a density unit. This S stands for a fancy name called Svedberg. It doesn't mean that they're bad in math. 30 and 50 usually equal 80, but in this case equal 70. Joke never works. Nobody likes it. See, they put them together. They do that. We'll stop there. Have fun. See you next week.